Hey guys, welcome to our first lecture for chapter 4. We're into cells right now, which we knew we'd get there eventually. It's a biology class after all. We're not going to dwell on it terribly long because you guys have already learned about cells. We spent a lot of time on them in general biology, and so this is going to be kind of a refresher course on some stuff you've forgotten, in the year or two since you took Gen Bio. So hopefully it's a good time and we get to talk a little application, and let's just jump right into it here. So we're talking cells, and their first little introduction in your chapter is your food for thought. They talk about E. coli. And so microorganisms, so microbes, little tiny cells, live on and in the human body, and they outnumber our own cells 10 to 1, which is kind of a weird thing to think about. But if you think about little bacteria on your skin and little bacteria in your gut, there are more things living on you and in you than there are cells that make you you. So it's a little disturbing, but bear in mind that the cells that make up bacteria are, of course, very, very small. So we're not just, a huge film of bacteria, but we are a little bit, I guess, especially on the inside. So here we go. So their examples: E. coli, Escherichia coli, and it's a super common intestinal bacteria, and it's in cattle, and it's in sheep, and it's in all sorts of animals. And in their intestines, it doesn't really seem to bother them. It's probably serving a purpose for them, a good purpose with digestion or something along those lines. Where it becomes a problem for humans is when you get the right bacteria in the wrong place. For example, in humans, E. coli is not so good for us. And so the example they give you is fresh fruits and vegetables. You've probably heard news stories as for eggs and salads and that kind of stuff causing E. coli outbreaks. And so, somebody goes to a restaurant and they eat meat or a salad or something that's been contaminated with feces from one of these animals and wasn't cooked to, 160, 170 degrees Fahrenheit to kill the bacteria. They get the living bacteria in them, they're infected, and they are very, very sick. So the outbreaks cause illness, of course, and they also cause economic impact because all of those people at home puking their guts out aren't going to work. They're going to the hospital, which is stressing the hospital resources, and you kind of get the idea. Just a lot of people getting sick in one area. So there's E. coli, a picture of E. coli on a leaf. If this were E. coli on a steak, and you asked for your steak medium well, probably wouldn't be an issue because the heat would kill all of these little guys. But when it's E. coli on a salad and you just throw it down on the plate cold and eat it, then they're all going to get in your gut and live in you, and it's just going to be terrible. So there's E. coli. A pleasant thought to start the chapter off. So what's a cell? You guys know what cells are. You probably learned in Gen Bio they're like the smallest unit of life or something like that. So first guy to see them, Antony van Leeuwenhoek. The guy who actually gave him the name cells was Robert Koch. It reminded him of like little cubicles that monks would work in. He called those cells. And so he kind of got the idea when he looked at plant cells, like a really thin slice of cork. He was able to see these little boxes, which we know, of course, are cell walls, but at that time they didn't know what they were. So he called them cells. We were kind of getting the idea that there were small living things around us. And in biology, we define cells as the smallest unit of life. We will learn about stuff, and we already have learned about stuff that's smaller than a cell, but it doesn't fulfill all of our criteria for saying something is alive. So cell is the smallest unit of life. So the foundation for modern biology really is the cell theory. That's why it gets so much attention in your high school and your first year college biology courses. Cells are super important. So here's your cell theory. If you didn't already know it, you're going to know it now. 4 points to the cell theory. Number 1, everything that's alive has to consist of one or more cells. So you and I are living, we consist of cells. An E. coli bacterium is living, it consists of one cell. A virus is not living, it consists of no cells. Cool? Dogs, cats, fish, plants, one or more cells, alive. Teeny tiny amoeba cruising in the lake, one or more cells, it's alive. The cell is a structural and functional unit of all organisms. So that's point 2. A cell is the smallest unit of life. Individually, they are alive. even if they're part of a multi-celled organism. You and I are, of course, a multi-celled organism. And if I get a cut and I kill some skin cells, those individual skin cells are no longer alive. But the whole organism is. And vice versa, those little skin cells are each individually alive. Oh, sorry about that. So, all living cells arise by the division of pre-existing cells. So for young Christian men and women, this is kind of a weird point for you because you're thinking, wait a second, if everything evolved from nothing, then where'd the first cell come from? Might be a question, and we'll talk a little bit about that. But for the cell theory, all living cells arise from pre-existing cells. So one cell divides, cuts itself in half, and becomes two cells. Cells contain hereditary material, this point 4, which you guys know as DNA, and they pass that DNA on to their offspring when they divide. And so those are the 4 basic principles that we're going to build everything we know about cells off of those 4 principles. So no matter if we're talking about a hair cell in a human or a bacteria or an amoeba or, a intestinal cell in a dog, they're all going to have the same features, a few features in common. The first is a plasma membrane. You guys might have learned this as a cell membrane, and all it is a barrier. It separates the inside of the cell from the outside of the cell. It does get a little more fancy than that, but for now, just know that it's a barrier from the outside of the cell to the inside of the cell. It controls exchanges between the cell and its environment. It lets certain things in, it lets certain things out, that kind of stuff. It's important to have that. Cells also have the second thing they have in common is a cytoplasm. A cytoplasm is a jelly-like mixture of water, sugars, ions, and proteins, and all the cellular components inside the cell. And so the cytoplasm is just everything in the plasma membrane. Organelles, the cytosol that they're floating in, all that kind of stuff is a cytoplasm. So all cells have to have a membrane and then some contents within the inside. And then within those contents on the inside, they must have the third point, which is DNA. We have to have DNA because that's the only way we can make other cells that are like us. And so that's the hereditary material of cells. When a cell divides, prior to its division, it will duplicate all of its DNA so that it has extra copies of everything, and then it'll go ahead and divide. And there are some DNA in a few other locations other than just the nucleus that you're most familiar with, and we'll talk about those a little later too. So all cells, no matter what it is, you got to have a plasma membrane, you got to have a cytoplasm, you got to have DNA if you're going to be a cell, and everything we're going to talk about has those. So here's a few different cells. We've got a teeny tiny bacteria, a plant cell, and an animal cell. And you can see just from looking across, they're all pretty different. But going back to that last point that we have a plasma membrane on a bacteria, a plasma membrane on a plant, and a plasma membrane on an animal. We also have a cytoplasm, so there's some stuff inside this, there's some stuff inside this, and there's some stuff inside this. Are they different? Sure, but there's something inside of them. And then finally, DNA. Bacteria, it's not too fancy. It's just DNA in there. Here we have DNA in our plant cell, and here we have DNA in our animal cell. So we can look at all of these and say, well, they're very different, and they behave differently, and they're found in different places. These are all cells, and they all have what they need to be a cell. So there's a few different cells. One question that you might have come up with in the past, and if you haven't, you can certainly talk about it now, is why are cells so small? Why isn't there an animal in the world that's just like one big cell the size of a table, that crawls around in a swamp somewhere? Where's the giant cell? Where's the cell that's like the size of a basketball, and there's not. And the reason there's not is because cells have constraints on how big they can be. And what it comes down to is what's called surface area to volume ratio. And this restricts the size of our cells. Here we have 2 different sized spheres, and you can think of them as, perhaps like a basketball or something that's filled with air. You know, a little beach ball filled with air, and then a bigger beach ball filled with air, and then the biggest beach ball filled with air. And you can see as we increase their diameter, it's this kind of linear increase, we increase their surface area, which is how much outside surface is available on the sphere. And we also increase their volume. And the volume is how much air would we have to pump into this beach ball to have it fully inflated. And what you see is as a beach ball, gets really large, its surface area catches up to its volume. So here I have the same amount of area on the surface of this beach ball as I do air inside the beach ball. Now, if I were working with a very small beach ball, I have 3 times the surface of the beach ball compared to the amount of air I can actually pump into that beach ball. Now, this is actually a good thing. It's good to have a high surface area compared to the volume of your interior for cells. Why do you think cells would want to have a high surface area to volume ratio? Well, our answer is that cells aren't beach balls. They're not just filled with air. They are filled with sugars and proteins and carbohydrates and lipids, and they make wastes, and those wastes have got to be exported. And they also need ions and sugars and food and energy, and they've got to bring in those sorts of gases and lipids into the cell, and they've got to transport it around inside the cell. And so what you get when you look at a cell that's just massive is it's so big and it has such a low surface area to volume ratio that it basically can't move its waste products out fast enough. For him to move waste products out is a very easy job. For him, it's almost impossible compared to how quickly he'll be producing the waste products. And so really big cells can't do their jobs very well because they have a small surface area and they will accumulate toxins. They can't move food as far as they need to move it. And so cells are small because they maintain that surface area to volume ratio. And for you and I, pardon me, when we look at things like production of enzymes and digestion, our cells and our intestines and stomach and things like that, having this really high surface area to volume ratio makes them so much better at their job. So that's one cool way to look at the size of cells. So how do we see cells? Not a surprise to you guys at this point in your life. You use a microscope. Most of these cells are super tiny, 10 to 20 micrometers. You can't really see them with the naked eye. There's a few big cells, big protists that you can, but for the stuff that makes up you and I, we really can't see most of this with the naked eye. So 1 micrometer is 1,000th of a millimeter, which is 1,000th of a meter. So we're talking real small here. Here's a kind of give you an idea. We're talking micrometers, so what you'd be trying to look at is 0.000004 inches. That's very, very, very small to give you an idea of how big most cells are. So if we want to look at cells, you and I are going to use a light microscope because it's easy to use and we can afford it. Light microscopes use visible light, so there's honestly a little light bulb under the microscope and it shines up through the sample. And then you have a couple of lenses that focus the light into an eyepiece and you can see the cells. Some of the universities and folks who really have some time and money on their hands use electron microscopes. And light microscopes have a limit to how small they can look at something because the light rays have to bend in order to give you that image. And when you get an image that's so small that it doesn't mess with the light rays much, you need a smaller ray. So these guys use a ray of electrons, and their wavelengths are super tiny. And so these guys will bend around super small samples. And so they can take pictures of stuff like proteins and lipids and that kind of stuff, which is amazing. The bad thing about an electron microscope is your sample pretty much has to be dead. If it's not, it's gonna be. And you're not going to get any color. Some simple physics tells you light waves make color. So if I'm smaller than light waves, this is going to be a black and white image. But in your book, they'll add color in, after the fact to give these really cool pictures of all sorts of stuff. So there's 2 types, an SEM and a TEM. An SEM is a scanning electron microscope, and it's only going to give you the image of the surface. And so we bounce electrons off the sample, and you can see the surface of a cell, or you can see the surface of something else, a protein, or the surface of a virus would be a good one, the surface of a virus. Then you have transmission, where we look at the inside of something. So we got to pass electrons through the sample, and we can get pictures of what's on the inside of this cell. And so these have really moved microscopy really in amazing directions now, because the things that we can take pictures of are so much smaller than we ever would have imagined, 50 or 60 years ago. So it's kind of a cool deal. So here's kind of the scale that we're looking at. This is a couple meters tall. As you move down to about frog eggs and what's this? Some sort of aquatic nymph stage of an insect. That stuff you can see with the naked eye right here. Down to the really small insects in rivers and stuff, we're not going to quite be able to see them with the naked eye. And we're going to get into most eukaryotic cells, which we really can't see with the naked eye. Bacteria, we can't see them, but we can see them with a light microscope. And then little mitochondria in cells. You can see a few of those, but you're going to be straining your eyes pretty hard at this point. So organelles inside the cells where light microscopy ends and electron microscopy begins. So electron is going to go all the way down to like big molecules. They can almost take a picture of an atom, as hard as that is to believe. So lipids, carbs, proteins, DNA, all the way up to viruses, and then really incredible pictures of mitochondria and bacteria for electron microscopy. And so there's kind of give you an idea of sort of where we fit with the whole scheme of size of organisms and structures we can look at. So prokaryotes. We got to separate all the cells. We're going to talk about this chapter into two sections. There's prokaryotes, which is before the nucleus, and there's eukaryotes, which is after the nucleus. And so we're starting with prokaryotes. Think simple here. No nucleus. These are bacteria and archaea. If you don't already know, archaea are just really tough bacteria that live in really nasty environments like hot springs and salty environments and boiling sulfur vents in the ocean, those kind of gross places. And so really small, really simple critters with no nucleus. One cell, that's it. The whole organism is one cell. And these are the smallest and most diverse life forms that we know of. There are a lot of different types of bacteria, and they share some pretty basic structures in really all the prokaryotes that we're going to look at. And so here's your typical bacteria. Not to say that all bacteria have this sort of stuff, but for the sake of labeling, we throw them on there. Flagella, well, it's one. Flagellum is singular, flagella is plural, is this long whip-like tail, and it's going to basically just whip around in this liquid media and push the bacteria around like a little propeller. They have a pilus, and the pilus can help them stick to structures, and it can also be used in conjugation. They can kind of stick a pilus into their neighbor and send over a plasmid, which is kind of cool. So let's see, we have a capsule, which not all bacteria have this, but the ones that do are encapsulated bacteria. Think of it as just another layer of protection against the harsh environment that they live in. It's going to help them, in some cases, infect certain animals when they couldn't before. Capsules are good for that. Inside the capsule is a cell wall. Not like a plant cell wall necessarily, but similar in its, function. And then inside of the cell wall, we have a plasma membrane, and the plasma membrane is the innermost layer of protective covering. And so they have a lot of protective covering. Capsule, cell wall, cell membrane. And once we're in a cell membrane, we're in a cytoplasm. So inside the cytoplasm, these guys do not have a nucleus. We said that, but they do have what's called a nucleoid. And so a nucleoid is a region of the bacteria where most of the DNA, pardon me, I'm drying out a little bit, where most of the DNA is gathered. So think about it like if the bacteria is a big plate and it's got spaghetti on the plate, you just scrape all that spaghetti to one side and you call it good enough. That's what they do with DNA. They move all their DNA over here and they say, yep, that's what we can do with it. There's the DNA. We can't put it in a nucleus, nothing fancy, but it's there. And then out here you have the cytoplasm, which is, our cytosol, our fluid, this stuff's floating in. These little green dots are ribosomes to make proteins. And that's about it. Like, this guy lives a simple life. He really does. He's got DNA, to pass on to the next cell he divides into. He's got ribosomes to make proteins. He uses the proteins to make his own capsule and his pilus or pili, and then he can swim around his media, and that's like it. That's his whole existence. It's pretty simple, but there's a lot of different types of bacteria, and they are very important. So here you have 'em. If you didn't get a chance to write them down because I was talking so fast, now's the time. Cell wall surrounds the plasma membrane. It's made of what's called peptidoglycan, or a variety of proteins in archaea. Peptidoglycan is super important because it allows us to target bacterial cells and kill them with antibiotics without killing the person who's sick, which is really nice. A lot of them are coated with a sticky capsule. It helps them stay to their environment and also helps them infect certain hosts. And then they have a flagellum, which helps them move around, and then pili, which can move the cells across the surface or sometimes be used for bacterial sex, where they extend a sex pilus and then they send over a copy of a plasmid. It's nothing really like vertebrate reproduction. They just call it sex because it's an exchange of DNA. So don't get too confused there. Ribosomes, these organelles, are where we assemble polypeptides, which really for these guys pretty much just means protein. That's all it means. They build proteins. That's about it. The nucleoid, all of our DNA is shoved into one region we call the nucleoid. And then plasmids are small circles of DNA that really only carry a few genes. There's no strict location for them. They just kind of hang out to our knowledge. And some plasmids carry a few genes that might not really make much of a difference at all. Others are incredibly important. So it can vary, but plasmids are something that are pretty unique to really just our bacteria. You and I, we don't have plasmids, at least not to my knowledge. I hope we don't. But there you have it. That's something that bacteria have that we don't. So biofilms is kind of a cool example of bacteria sort of living together. Even though all our prokaryotes are single-celled, they just live as one cell, that doesn't necessarily mean that they just live by themselves away from all other bacteria. Your book gives us this idea of a biofilm. And a biofilm, just think slimy bacteria-filled residue on old jelly or a toilet or a locker or something like that. All these single-celled organisms live together and they share this layer of polysaccharides and glycoproteins that they all live on together. And so they kind of secrete their own, I don't know, house, neighborhood, and then they all live there together on that film of sugar protein stuff. And you could have bacteria living with other bacteria. There could be algae in the mix, fungi, protists, even archaea living there. And it's kind of a cool idea because what biofilms allow for is it's sort of like a really small ecosystem that's living between, I don't know, your teeth or something like that. And biofilms are neat because as these little tiny ecosystems get built up on this little layer of slime, it allows bacteria that are weaker and couldn't normally survive in that environment to be able to survive in that environment. And so you get this really teeny tiny, just microcosm view of an ecosystem. But it's just bacteria in the ecosystem. And when they all live together, they're more biodiverse and they're more successful when they have multiple types of species and bacteria living together. And so it's kind of cool to see, this big concept that we apply to a place like, I don't know, Yellowstone National Park, where, we want this whole complete ecosystem with every little niche filled, and we take that same concept and we can actually apply it to a little yellow spot, between our teeth. The same exact thing is happening. So it's kind of a big idea for you. It's a little weird, but there you have it. So there is dental plaque. Those hard to reach places build up a plaque, and that plaque is a substrate for a biofilm to grow on. And here you can see, there's a rod-shaped bacteria, there's some circular bacteria, there's some big circular bacteria, and then this purple spot, it could be the biofilm or it could be super thin strands of bacteria. My guess is it's probably the biofilm because it's significantly smaller even than these little coccus bacteria here. So there you have it. You've got what, 3 or 4 species living here together, forming this biofilm on teeth. And of course, that ends up not being so good for teeth because they'll eventually wear away the enamel, and then it leads for a site to infection and that kind of stuff. So it's not so great for your teeth, but it is great to understand that these guys live together in ways that are very similar to the big ecosystems that we understand well. So we're gonna stop there. I'm going to show you a video that takes you through a prokaryotic cell so you get a break from my voice, and we're going to call it good to go for today. Let's see. There we are, prokaryote. Got our volume up?
Prokaryotes are the smallest known cells and the simplest in structure. The bacterial chromosome is a single circular molecule of DNA. It occupies a region of the cytoplasm called the nucleoid. The tiny volume of cytoplasm is continuous with the region of DNA. Ribosomes give the cytoplasm a grainy appearance. They are the molecular machinery upon which polypeptide chains are assembled. The plasma membrane mediates the flow of substances to and from the cytoplasm. It has protein channels, transporters, and receptors for signals and substances. Most bacteria have a semi-rigid or rigid cell wall that surrounds the plasma membrane, supports the cell, and imparts shape to it. A thick, protective capsule of jelly-like polysaccharide is the outermost layer of many bacteria. Pili are protein filaments that help cells attach to various surfaces and to one another. Bacterial flagella allow some prokaryotic cells to move about in a fluid environment.
Okay, boy, I don't know if that was better than my voice or not, but there you have it. Somebody other than me talking you through this. So we're going to call it good with prokaryotes for there, and I appreciate you guys' time. Thank you.